Limb development and regeneration in vertebrates are remarkable processes involving complex interactions between different tissues, signaling pathways, and genetic factors. The development of limbs from the embryonic stage to adulthood is crucial for the proper formation of functional appendages in vertebrates, including humans. Additionally, the ability of certain vertebrates, such as amphibians, to regenerate lost limbs presents a fascinating biological phenomenon that is still a subject of intense scientific investigation. Understanding these processes has significant implications for developmental biology, regenerative medicine, and the study of limb malformations and amputations. Limb development in vertebrates. The development of limbs in vertebrates is a highly regulated process that involves the formation of distinct tissues, such as muscle, bone, nerves, and blood vessels, which all contribute to the final structure and function of the limb. Limb development in vertebrates occurs in a series of well-defined stages and is controlled by several key signaling pathways, including FGF fibroblast growth factor, sonic hedgehog, and WNT signaling. This section explores the steps of limb development in detail. 1. Early limb bud formation. Limb development begins at an early embryonic stage, with the initiation of limb buds from the lateral plate mesoderm, which gives rise to the mesodermal progenitors of bones and muscles. The process begins with the formation of the limb field, regions of the embryonic body where limbs will develop. These fields are specified early in development and include cells that will give rise to the forelimbs and hind limbs. The signaling pathway that drives limb bud formation includes the FGF10 signaling from the lateral plate mesoderm, which activates the overlying ectoderm to form the apical ectodermal ridge hair. The hair is essential for the elongation and proper development of the limb bud, as it secretes FGF8 and FGF4, which are crucial for the outgrowth of the limb. 2. Limb bud patterning. Once the limb bud forms, it undergoes a process known as limb patterning. This involves the establishment of the proximal distal axis from the body to the tip of the limb, the anterior posterior axis from the thumb to the little finger, and the dorsal ventral axis, the back to palmar surface. Proximal distal patterning. The hair is crucial for regulating the growth of the limb along this axis by secreting FGFs. The zone of polarizing activity, ZPA, at the posterior tip of the limb bud secretes sonic hedgehog, a key signaling molecule that regulates proximal distal growth. Anterior posterior patterning. The hair and ZPA interact to control the proper positioning of digits in the limb. The SHISH signaling gradient from the posterior region helps define the patterning of digits. Dorsal ventral patterning. The dorsal ventral axis is controlled by the interaction of WNT and BMP signaling. The dorsal side of the limb expresses WNT7A, while the ventral side expresses GRAIL1, EN1, which serves as an inhibitor of WNT signaling. 3. Limb development and differentiation. Once patterning is established, limb mesodermal cells begin to differentiate into cartilage and bone. The stylopod upper arm or thigh, zygopod forearm or shank, and autopod hand or foot are defined along the proximal distal axis. Chondrocytes in the limb mesoderm differentiate into cartilage, which later ossifies to form bones. The differentiation of muscle precursors into skeletal muscle also occurs at this stage, under the influence of myogenic regulatory factors, MRFs. In parallel, nerves from the spinal cord extend into the developing limb and innervate muscles, which is crucial for coordinated limb movements. The blood vessels also develop and establish circulation within the limb. Limb regeneration in vertebrates. While most adult vertebrates have limited regenerative capacity, certain species, particularly amphibians, exhibit remarkable regenerative abilities. Limb regeneration involves the ability to regrow a fully functional limb after amputation. The mechanisms of limb regeneration have been a focus of extensive research, and although much has been learned, the complete process is still not fully understood. 1. The regeneration process in amphibians, e.g., salamanders. In amphibians like salamanders, limb regeneration occurs in several well-characterized stages. Wound healing. After amputation, the first response is wound closure, which is followed by the formation of a regenerative blastema, a mass of undifferentiated cells at the site of injury. This is a crucial step in regeneration. Differentiation and proliferation. Cells in the amputation site undergo differentiation, reverting to a more primitive state and acquiring the ability to proliferate. These cells then become part of the blastema, where they will proliferate and differentiate into the various tissues needed for limb regeneration. Patterning and differentiation. The regenerative blastema undergoes patterning to form the basic structure of the limb. This involves signaling molecules such as FGFs, BMPs, SHISH, and WNTs. The limb regenerates from the distal end to the proximal region, re-establishing the correct structure, including bone, muscle, and nerves. Reinnervation and functional recovery. Nerve innervation is essential for proper regeneration. The reinnervation of the regenerated limb by sensory and motor neurons helps in the functional recovery of the limb. 2. Regeneration in mammals. In contrast to amphibians, most mammals, including humans, do not have the ability to regenerate entire limbs. However, there is some regenerative potential in certain tissues, such as the digit tips and liver. In mammals, limb regeneration is limited to the formation of a scar rather than a fully functional new limb. There has been ongoing research aimed at understanding the mechanisms that allow amphibians to regenerate limbs, with the hope of applying this knowledge to mammalian regenerative medicine. Scientists are particularly interested in the role of stem cells in differentiation in limb regeneration. Molecular signaling in limb regeneration. Key molecular signaling pathways involved in limb regeneration include FGF signaling. FGFs play a crucial role in both limb development and regeneration. During regeneration, FGF2 is essential for promoting the growth and proliferation of cells in the blastema. SHISH signaling. Sonic hedgehog signaling is crucial for maintaining proper patterning during both limb development and regeneration. SHISH gradients are important for the proximal distal patterning of the regenerated limb. WNT signaling. WNT signaling helps regulate stem cell proliferation and differentiation during regeneration. Inhibiting WNT signaling has been shown to prevent limb regeneration in amphibians. BMP signaling. Bone morphogenic proteins, BMPs, help control the differentiation of mesodermal cells into bone and cartilage. Modulating BMP signaling can enhance or inhibit limb regeneration, depending on the timing and context. CSIRNet JRF Life Sciences. Previous year questions on limb development and regeneration. CSIRNet JRF June 2019. Which signaling pathway is crucial for proximal distal patterning during limb development in vertebrates? Answer. FGF signaling. Explanation. The air apical ectodermal ridge releases FGF signals that regulate limb outgrowth along the proximal distal axis. CSIRNet JRF December 2018. In salamanders, which structure is essential for the regeneration of limb? Answer. Blastema. Explanation. The blastema is a mass of undifferentiated cells that forms at the site of injury and is crucial for limb regeneration. CSIRNet JRF June 2017. What is the role of sonic hedgehog SHISH in limb development? Answer. Sonic hedgehog SHISH signaling is responsible for the patterning of the anterior posterior axis during limb development, influencing digit formation and growth. Explanation. SHISH gradients control the patterning and differentiation of tissues in the developing limb. Conclusion. The development and regeneration of limbs in vertebrates represent two of the most fascinating processes. Limb development involves a well-coordinated series of events regulated by various signaling pathways, ensuring the proper formation of functional limbs. In contrast, the regenerative ability seen in amphibians highlights the potential for tissue regeneration, a process that, although absent in most mammals, has significant implications for regenerative medicine. Understanding the molecular and cellular mechanisms underlying both processes could open new avenues for treating limb injuries and congenital defects. You said. Differentiation of neurons. ChatGPT said. Differentiation of neurons. CSIRNet JRF Life Sciences lecture. The differentiation of neurons is a critical process in the development of the nervous system, allowing for the formation of specialized cells capable of transmitting electrical signals. This process involves the transformation of precursor cells, also known as neural progenitors, into fully differentiated neurons, which is regulated by a combination of intrinsic genetic programs and extrinsic signals from the surrounding environment. Understanding the molecular pathways and mechanisms involved in neuronal differentiation is crucial for grasping how the nervous system develops, how it is maintained, and how it can potentially regenerate after injury. 1. Early stages of neural development. Neuronal differentiation begins early during embryonic development from a common pool of neural progenitor cells that originate from the plate. The neural plate itself arises from the ectoderm, the outermost layer of the embryo. The neural progenitors are initially pluripotent, meaning they have the potential to differentiate into any type of neural cell, including neurons, glial cells like astrocytes and oligodendrocytes, and ependymal cells. The first critical step in neural differentiation is the process of neural induction, where specific signaling molecules from surrounding tissues like the notochord and somites cause the ectodermal cells to differentiate into neural progenitor cells. This is primarily regulated by BMP, bone morphogenic protein antagonists, such as noggin and chordin, which block the BMP signaling pathway, preventing the ectoderm from becoming skin and instead promoting the formation of the neural plate. 2. Formation of the neural tube. Once the neural plate forms, it folds to give rise to the neural tube, a structure that will eventually develop into the central nervous system (CNS), including the brain and spinal cord. As the neural tube closes, cells along the neural tube differentiate into various types of neural progenitors. These progenitors will eventually give rise to neurons, astrocytes, oligodendrocytes, and ependymal cells, each serving specific roles in the nervous system. 3. Neural stem cells in the neural progenitor pool. Neural stem cells (NSCs) in the developing embryo are self-renewing, multipotent cells capable of producing both neurons and glial cells. During neurogenesis, the formation of new neurons, the NSCs undergo a process of asymmetric cell division, where one daughter cell remains a stem cell and the other becomes a neural progenitor cell, which is committed to differentiating into a specific type of neural cell. The fate of the neural progenitor cell is determined by a combination of intrinsic genetic programs and extrinsic signals from the microenvironment. As the progenitor cells begin to exit the cell cycle, they start to differentiate into postmitotic neurons. This transition involves significant changes in gene expression, which will be discussed further below. 4. Molecular mechanisms of neuronal differentiation. The differentiation of neurons is regulated by several signaling pathways and transcription factors that orchestrate the precise timing and identity of neurons. Some of the key molecular players involved in neuronal differentiation include A. Notch signaling pathway. The Notch signaling pathway plays a critical role in regulating the differentiation of neural progenitors. Notch signaling helps maintain progenitor cells in an indifferentiated state. When Notch receptors are activated by ligands from adjacent cells like delta and jagged, it inhibits the differentiation of progenitor cells, maintaining them in a proliferative state. Conversely, the inhibition of Notch signaling promotes differentiation into neurons. This pathway helps balance the number of progenitor cells and differentiated neurons. B. Basic helix loop helix (VHLH) transcription factors. A family of transcription factors known as VHLH proteins plays a central role in neuronal differentiation. The neurogenin NGN and neurod proteins, which are members of the VHLH family, are key regulators of neuronal fate. Neurogenin 1 NGN1 and neurogenin 2 NGN2 are expressed in the neural progenitors and promote the differentiation of neurons by activating the expression of neuronal genes. Neurod further promotes differentiation into specific neuronal types, ensuring proper neuronal identity. C. Sonic hedgehog SHISH signaling. The SHISH signaling pathway, originally discovered in the context of limb development, also plays an important role in neuronal differentiation. SHISH is secreted by the notochord and floor plate in the spinal cord and is essential for the patterning of the neural tube. SHISH regulates the ventral patterning of the spinal cord, where it helps define the motor neuron pool and the identity of other neuronal subtypes. In the brain, SHISH signaling contributes to the differentiation of specific neuronal populations, such as those in the ventral forebrain. D. WNT signaling. The WNT signaling pathway is involved in both the development and differentiation of neurons. WNT signaling regulates the decision between neurogenesis and gliogenesis, the formation of glial cells. WNT inhibitors are essential for ensuring that progenitor cells exit the cell cycle and differentiate into neurons, while WNT activators are involved in promoting gliogenesis. WNT signaling is also crucial for the maintenance of the neural stem cell pool during development. E. Retinoic acid signaling. Retinoic acid, a derivative of vitamin A, acts as a potent signaling molecule that influences neuronal differentiation. During neurogenesis, retinoic acid regulates the expression of genes involved in neuronal differentiation and axonal outgrowth. It is particularly important in the hindbrain, where it helps define the identity of neurons in specific regions. 5. Synaptic development and neuronal maturation. After the initial differentiation into neurons, the next step is the formation of synaptic connections, which are crucial for neuronal function. Neurons extend axons to specific target regions, where they establish synaptic contacts with other neurons or effector cells, such as muscle cells. The formation of synapses involves the release of neurotrophic factors, which promote the growth and survival of neurons. The differentiation of neurons is a critical process in the development of the nervous system, allowing for the formation of specialized cells capable of transmitting electrical signals. This process involves the transformation of precursor cells, also known as neural progenitors, into fully differentiated neurons, which is regulated by a combination of intrinsic genetic programs and extrinsic signals from the surrounding environment. Understanding the molecular pathways and mechanisms involved in neuronal differentiation is crucial for grasping how the nervous system develops, how it is maintained, and how it can potentially regenerate after injury. 1. Early stages of neural development. Neuronal differentiation begins early during embryonic development from a common pool of neural progenitor cells that originate from the neural plate. The neural plate itself arises from the ectoderm, the outermost layer of the embryo. The neural progenitors are initially pluripotent. The neural progenitors are initially pluripotent, meaning they have the potential to differentiate into any type of neural cell, including neurons, glial cells like astrocytes and oligodendrocytes, and ependymal cells. The first critical step in neural differentiation is the process of neural induction, where specific signaling molecules from surrounding tissues like the notochord and somites cause the ectodermal cells to differentiate into neural progenitor cells. This is primarily regulated by BMP, bone morphogenic protein antagonists, such as noggin and chordin, which block the BMP signaling pathway, preventing the ectoderm from becoming skin and instead promoting the formation of the neural plate. 2. Formation of the neural tube. Once the neural plate forms, it folds to give rise to the neural tube, a structure that will eventually develop into the central nervous system (CNS), including the brain and spinal cord. As the neural tube closes, cells along the neural tube differentiate into various types of neural progenitors. These progenitors will eventually give rise to neurons, astrocytes, oligodendrocytes, and ependymal cells, each serving specific roles in the nervous system. 3. Neural stem cells in the neural progenitor pool. Neural stem cells (NSCs) in the developing embryo are self-renewing, multipotent cells capable of producing both neurons and cells. During neurogenesis, the formation of new neurons, the NSCs undergo a process of asymmetric cell division, where one daughter cell remains a stem cell and the other becomes a neural progenitor cell, which is committed to differentiating into a specific type of neural cell. The fate of the neural progenitor cell is determined by a combination of intrinsic genetic programs and extrinsic signals from the microenvironment. As the progenitor cells begin to exit the cell cycle, they start to differentiate into postmitotic neurons. This transition involves significant changes in gene expression, which will be discussed further below. 4. Molecular mechanisms of neuronal differentiation. The differentiation of neurons is regulated by several signaling pathways and transcription factors that orchestrate the precise timing and identity of neurons. Some of the key molecular players involved in neuronal differentiation include A. Notch signaling pathway. The Notch signaling pathway plays a critical role in regulating the differentiation of neural progenitors. Notch signaling helps maintain progenitor cells in an indifferentiated state. When Notch receptors are activated by ligands from adjacent cells, like delta and jagged, it inhibits the differentiation of progenitor cells, maintaining them in a proliferative state. Conversely, the inhibition of Notch signaling promotes differentiation into neurons. This pathway helps balance the number of progenitor cells and differentiated neurons. B. Basic helix loop helix BHLH transcription factors. A family of transcription factors known as BHLH proteins plays a central role in neuronal differentiation. The neurogenin NGN and neurod proteins, which are members of the BHLH family, are key regulators of neuronal fate. Neurogenin 1 NGN1 and neurogenin 2 NGN2 are expressed in the neural progenitors and promote the differentiation of neurons by activating the expression of neuronal genes. Neurod further promotes differentiation into specific neuronal types, ensuring proper neuronal identity. C. Sonic hedgehog SHISH signaling. The SHISH signaling pathway, originally discovered in the context of limb development, also plays an important role in neuronal differentiation. SHISH is secreted by the notochord and floor plate in the spinal cord and is essential for the patterning of the neural tube. SHISH regulates the ventral patterning of the spinal cord, where it helps define the motor neuron pool and the identity of other neuronal subtypes. In the brain, SHISH signaling contributes to the differentiation of specific neuronal populations, such as those in the ventral forebrain. D. WNT signaling. The WNT signaling pathway is involved in both the development and differentiation of neurons. WNT signaling regulates the decision between neurogenesis and gliogenesis, the formation of glial cells. WNT inhibitors are essential for ensuring that progenitor cells exit the cell cycle and differentiate into neurons, while WNT activators are involved in promoting gliogenesis. WNT signaling is also crucial for the maintenance of the neural stem cell pool during development. E. Retinoic acid signaling. Retinoic acid, a derivative of vitamin A, acts as a potent signaling molecule that influences neuronal differentiation. During neurogenesis, retinoic acid regulates the expression of genes involved in neuronal differentiation and axonal outgrowth. It is particularly important in the hindbrain, where it helps define the identity of neurons in specific regions. 5. Synaptic development and neuronal maturation. After the initial differentiation into neurons, the next step is the formation of synaptic connections, which are crucial for neuronal function. Neurons extend axons to specific target regions, where they establish synaptic contacts with other neurons or effector cells, such as muscle cells. The formation of synapses involves the release of neurotrophic factors, which promote the growth and survival of neurons. Synaptogenesis involves the clustering of receptors and ion channels at the postsynaptic site and the establishment of presynaptic boudins that release neurotransmitters. The maturation of these synapses is critical for establishing functional neural circuits. 6. Role of extracellular matrix in neuronal differentiation. The extracellular matrix (ECM) plays a significant role in supporting and guiding neuronal differentiation. ECM proteins such as laminin, fibronectin, and collagens provide structural support and regulate cell migration, differentiation, and synaptogenesis. The interaction between integrins, cell surface receptors, and ECM proteins helps guide the differentiation of neurons and the formation of synaptic contacts. CSIR-NET JRF Life Sciences. Previous year questions on neuronal differentiation. CSIR-NET JRF June 2020. Which of the following signaling pathways is primarily involved in the differentiation of neural progenitors into neurons? Answer: Notch signaling. Explanation: Notch signaling is essential for maintaining neural progenitor cells in an undifferentiated state and inhibits premature neuronal differentiation. When Notch signaling is reduced, progenitor cells exit the cell cycle and differentiate into neurons. CSIR-NET JRF December 2018. Which transcription factor family is essential for promoting neuronal differentiation in neural progenitor cells? Answer: BHLH transcription factors. Explanation: The BHLH transcription factors, such as neurogenin and neurod, are key regulators of neuronal differentiation in neural progenitor cells. CSIR-NET JRF June 2017. What is the role of retinoic acid in neuronal differentiation? Answer: Retinoic acid promotes neuronal differentiation by regulating gene expression involved in neurogenesis and synaptic maturation. Explanation: Retinoic acid is a key factor in regulating neuronal differentiation, particularly in the hindbrain region. Conclusion: Neuronal differentiation is a highly coordinated process, regulated by multiple signaling pathways, transcription factors, and environmental cues. The understanding of how neural progenitors give rise to functional neurons not only informs our knowledge of embryonic development but also has implications for regenerative medicine, particularly in the context of neurodegenerative diseases and spinal cord injuries. The ongoing study of molecular signals and mechanisms involved in neuronal differentiation promises to yield insights into the complexities of the nervous system and how it can potentially be repaired. You said. Post-embryonic development larval formation. ChatGPT said. Post-embryonic development, larval formation, CSIR-NET JRF Life Sciences lecture. Post-embryonic development refers to the growth and differentiation processes that occur after the formation of the embryo, eventually leading to the development of a fully functional organism. In many animals, especially in those that undergo indirect development metamorphosis, the embryonic stage gives rise to a larval stage. The larval stage serves as a transitional phase before the organism undergoes metamorphosis and assumes its adult form. Understanding larval formation is crucial for gaining insights into developmental biology, as it showcases the diversity of developmental strategies and the regulation of gene expression that shapes an organism's life cycle. 1. Larval formation. An overview. The larval stage is an intermediary form of development found in many invertebrates and some vertebrates. It is typically different from the adult form in terms of morphology, physiology, and behavior. The larva often serves to exploit different ecological niches from the adult, with the purpose of feeding, growth, or dispersion. Over time, the larva will undergo metamorphosis, transforming into the adult form. In species that exhibit indirect development, such as most arthropods, amphibians, and some fish species, larvae are produced after fertilization and hatching. These larvae typically undergo dramatic changes during their development, a process known as metamorphosis, which involves extensive remodeling of tissues and organs to transition into the adult form. 2. The larval stage in development. The larval stage represents a critical developmental phase and exhibits several key characteristics. Morphological differences. Larvae often look significantly different from adults, having distinct features such as different body structures, number of segments, and appendages. This differentiation allows larvae to occupy specific ecological niches that the adult form may not exploit. Feeding and growth. In most species, larvae are highly specialized for feeding and growth. Their feeding apparatus, digestive system, and behavior are adapted to maximize energy intake, facilitating their rapid growth to support the next stage of development. Locomotion. The method of locomotion in larvae differs from adults. For example, in amphibians, the tadpole larva swims using a tail, while the adult develops legs for terrestrial locomotion. Metamorphosis. Larvae undergo metamorphosis, a biological process that transforms the larval form into the adult form. This process involves significant physiological changes, including the reorganization of tissues, structures, and organs. 3. Types of larval forms. Different species exhibit different types of larvae based on their developmental strategy. Some key examples include A. Holometabolous metamorphosis, complete metamorphosis. In species exhibiting holometabolous metamorphosis, the larval form is quite distinct from the adult. This type of development is characterized by 4 major stages: egg right-pointing arrow larva, right-pointing arrow pupa, right-pointing arrow adult. During the larval stage, the organism is primarily in a feeding and growth phase, consuming resources to prepare for metamorphosis. Some well-known examples of animals with holometabolous development include butterflies, beetles, and flies. In these species, the larva is commonly referred to as a caterpillar, larva, or grub depending on the species. Example: in butterflies Lepidoptera, the caterpillar is a larval stage, which feeds voraciously and grows rapidly. After reaching a critical size, the caterpillar enters the pupal stage chrysalis, where significant reorganization occurs, resulting in the adult butterfly. B. Hemimetabolous metamorphosis, incomplete metamorphosis. In hemimetabolous metamorphosis, the developmental stages include egg right-pointing arrow nymph, right-pointing arrow adult. Unlike holometabolous insects, hemimetabolous insects do not undergo a pupal stage. The nymph larval stage closely resembles the adult but lacks fully developed wings and reproductive organs. Over successive molts, the nymph gradually matures into an adult. Examples of hemimetabolous development are seen in grasshoppers, cockroaches, and dragonflies. Example: in grasshoppers, the nymphs are miniature versions of adults, differing only in the lack of wings and reproductive structures. As they molt, they gradually develop these features, transforming into fully functional adult grasshoppers. E. Indirect development in amphibians. In amphibians, particularly frogs and toads, the larval stage is called a tadpole. The tadpole is aquatic and has a tail, gills, and no limbs. Over time, during metamorphosis, it loses its tail, develops limbs, and its respiratory system shifts from gills to lungs, adapting it for a terrestrial lifestyle as an adult. Example: in frogs, the development starts with a fertilized egg, which hatches into a tadpole. The tadpole undergoes metamorphosis, where it loses its tail, grows legs, and undergoes major changes in internal organs, such as the transition from gill breathing to lung breathing. 4. Larval formation and differentiation. The process of larval formation begins immediately after fertilization when the zygote undergoes a series of cell divisions called cleavage. This results in a blastula stage, followed by the formation of the gastrula, where the primary germ layers ectoderm, mesoderm, and endoderm are established. These layers will give rise to various tissues and organs in both the larval and adult forms. A. Gastrulation and formation of germ layers. The gastrulation process marks the formation of the primary germ layers that will later differentiate into various tissues. The ectoderm will form the nervous system and skin, the mesoderm will give rise to muscles, bones, and the circulatory system, and the endoderm will form the digestive and respiratory systems. These developmental steps are foundational for the emergence of specialized larval organs. B. Larval organogenesis. Organogenesis in the larval stage involves the differentiation of various organs that are necessary for the larva's survival. For instance, larvae of many species form specialized mouthparts, digestive systems, and sensory organs that help them feed and interact with their environment. 5. Mechanisms regulating larval formation. The differentiation of larval structures is driven by gene expression patterns regulated by developmental pathways, including WNT signaling, hedgehog signaling, and Notch signaling, among others. These pathways guide the differentiation of progenitor cells into the various cell types that make up the larval body. A. WNT signaling. WNT signaling plays a crucial role in determining the anterior-posterior axis of the larval body and is involved in the specification of the head and tail structures. B. Hedgehog signaling. Hedgehog signaling is involved in patterning the neural tube and ensuring the proper segmentation and differentiation of the larval body. C. Notch signaling. Notch signaling regulates the differentiation of specific cell types and ensures proper development of tissues during larval formation. 6. Larval maintenance and growth. Once larvae are formed, they begin feeding and growing, increasing in size to prepare for the next phase of development. The growth of larvae is largely driven by nutrient intake, which is essential for the accumulation of energy reserves needed for metamorphosis. During this time, the larva undergoes several molts, shedding its exoskeleton in arthropods or undergoing other forms of growth in amphibians and other vertebrates. 7. CSIR Net JRF Life Sciences. Previous year questions on larval formation. CSIR Net JRF June 2019. Which of the following is a characteristic feature of holometabolous metamorphosis? Answer: the larval form undergoes a pupal stage before becoming an adult. Explanation: holometabolous metamorphosis involves distinct stages where the organism passes through a larval stage, followed by a pupal stage, and finally emerges as an adult. CSIR Net JRF December 2018. In the amphibian frog, which of the following processes takes place during the transition from tadpole to adult? Answer: loss of tail, development of limbs, and change from gill to lung respiration. Explanation: during metamorphosis in amphibians, the tadpole undergoes significant morphological changes, including the loss of its tail and the development of limbs, while also transitioning from gill to lung respiration. CSIR Net JRF June 2017. Which signaling pathway is involved in regulating larval development in many species? Answer: WNT signaling. Explanation: WNT signaling is essential for the patterning of the larval body, including anterior-posterior axis specification and tissue differentiation. Conclusion: the larval stage represents a critical period in the life cycle of many organisms, bridging the gap between embryonic development and adulthood. It involves a complex series of events including morphological changes, organogenesis, and growth, all of which are tightly regulated by a variety of signaling pathways. Larval formation is not only an important developmental process but also a key point of study for understanding the broader principles of developmental biology and metamorphosis across diverse species. You said, metamorphosis, ChatGPT said. Metamorphosis, CSIR Net JRF Life Sciences Lecture. Metamorphosis is a pivotal process in the life cycle of many organisms, particularly those that undergo indirect development, such as insects, amphibians, and certain marine invertebrates. It refers to the drastic and often dramatic morphological, physiological, and behavioral changes that an organism undergoes as it transforms from a juvenile form, often very different in appearance and function from the adult, into a fully mature organism. Metamorphosis can be classified into two major types: complete metamorphosis holometabolism and incomplete metamorphosis hemimetabolism, each with distinct mechanisms and stages. 1. Overview of metamorphosis. Metamorphosis represents the transition from a larval or juvenile stage to an adult form. The term is often used in the context of species that undergo major transformations, which may involve the complete reorganization of the organism's body structure. Metamorphosis is regulated by intricate hormonal signaling pathways that orchestrate the timing and progression of developmental events. The process of metamorphosis is essential for species that require a juvenile phase to feed, grow, or disperse before assuming a sexually mature adult form. For example, in insects, metamorphosis allows the larval stage to exploit different ecological niches from the adult, ensuring the survival of the species by reducing competition between life stages. 2. Types of metamorphosis. A. Complete metamorphosis, holometabolism. In complete metamorphosis, the organism undergoes 4 distinct stages: egg, right-pointing arrow, larva, right-pointing arrow, pupa, right-pointing arrow, adult. The larval and adult stages differ significantly in both morphology and function. During the pupal stage, the organism undergoes significant internal reorganization and restructuring of tissues, resulting in the formation of adult characteristics. This type of metamorphosis is common in insects such as butterflies, beetles, and flies. Key stages in holometabolism. Egg stage. The fertilized egg hatches into a larva, often significantly different in form from the adult. Larval stage. The larva is primarily focused on feeding and growth. It is morphologically distinct from the adult and typically lacks wings and reproductive organs. For example, a caterpillar larva of a butterfly is structurally different from the adult butterfly. Pupal stage. During this stage, the larva undergoes metamorphosis, which may involve a resting or quiescent period. The tissues of the larva undergo extensive remodeling, resulting in the formation of adult structures such as wings, legs, and reproductive organs. This transformation occurs in a structure called the pupa, e.g., a chrysalis in butterflies. Adult stage. Once the organism emerges from the pupa, it is fully developed into an adult form with wings, reproductive organs, and other characteristics necessary for reproduction. Example: in butterflies, the caterpillar larva feeds and grows, then forms a chrysalis pupa, during which the internal tissues undergo reorganization to form the adult butterfly with wings. B. Incomplete metamorphosis, hemimetabolism. In incomplete metamorphosis, the organism progresses through 3 major stages: egg, right-pointing arrow, nymph, right-pointing arrow, adult. The nymph resembles a miniature version of the adult but lacks fully developed wings and reproductive organs. As the nymph molts, it gradually matures into the adult form. Unlike holometabolism, the nymph does not undergo a pupal stage, and the changes are more gradual. Key stages in hemimetabolism. Egg stage. The fertilized egg hatches into a nymph, which is similar in body structure to the adult but differs in size and the lack of certain adult characteristics, such as wings. Nymph stage. The nymph is a juvenile form that usually feeds and grows. It resembles the adult but lacks full morphological development, such as wings and genitalia. This stage is characterized by several molts, ecdysis, during which the nymph sheds its exoskeleton and increases in size. Adult stage. After several molts, the nymph matures into the adult form, now with wings in wing species and reproductive organs fully developed. Example: in grasshoppers, the nymph looks like a small adult grasshopper without wings and reproductive organs. Over time, as it molts, it develops wings and matures into an adult grasshopper capable of reproduction. 3. Hormonal regulation of metamorphosis. Metamorphosis is primarily regulated by hormones that control the timing and progression of developmental changes. The juvenile hormone, JH, and ecdysone are the 2 major hormones involved in regulating metamorphosis in insects. Juvenile hormone, JH plays a key role in maintaining the larval state. High levels of JH prevent the organism from entering the pupal or adult stage. As JH levels decrease, the transition to the next developmental stage is initiated. Ecdysone. Ecdysone is the hormone responsible for triggering molting and the progression through the metamorph stages. In holometabolous insects, the increase in ecdysone levels triggers the formation of the pupa, where a significant transformation occurs. In amphibians, the hormonal regulation involves thyroid hormones like thyroxine T4 and triiodothyronine T3, which initiate metamorphosis from the larval form tadpole into the adult form frog. The change is marked by the development of limbs, the loss of the tail, and the development of the lungs for terrestrial respiration. 4. Metamorphosis in amphibians. In amphibians, such as frogs and toads, metamorphosis from larval tadpole to adult frog is a classic example of vertebrate metamorphosis. During this process, tadpoles transition from an aquatic, gill-breathing stage to a terrestrial, lung-breathing adult. The development of legs, the loss of the tail, and the development of the digestive system for terrestrial feeding are hallmark changes. Key steps in amphibian metamorphosis. Gills to lungs. Tadpoles initially breathe through gills, but as metamorphosis progresses, they develop lungs for aerial respiration. Tail resorption. The tadpole tail is gradually resorbed as the adult form is established. Limbs development. Legs begin to emerge, and the tadpole acquires the ability to move on land. 5. Metamorphosis in other species. A. Marine invertebrates. In marine organisms like sea urchins and corals, metamorphosis is a crucial process for transitioning from a larval stage to the adult form. For example, in sea urchins, larvae undergo a metamorphosis that leads to the development of a spiny adult form, with a functional digestive system, arms, and reproductive organs. B. Insects and other arthropods. Insects such as beetles, flies, and moths undergo complete metamorphosis, whereas cockroaches and dragonflies exhibit incomplete metamorphosis. 6. CSIR Net JRF Life Sciences. Previous year questions on metamorphosis. CSIR Net JRF June 2019. What is the characteristic feature of holometabolous metamorphosis? Answer: the larva undergoes a pupal stage before becoming an adult. Explanation: in holometabolous metamorphosis, organisms transition through distinct stages, including a pupal phase, where significant internal reorganization occurs. CSIR Net JRF December 2018. Which of the following hormones plays a crucial role in the process of metamorphosis in insects? Answer: ecdysone. Explanation: ecdysone is the hormone that triggers molting and developmental changes during metamorphosis in insects. CSIR Net JRF June 2017. During metamorphosis in amphibians, what is the main organ that undergoes functional changes? Answer: the respiratory system, transition from gills to lungs. Explanation: the amphibian's respiratory system undergoes a major transformation during metamorphosis, where the gills of the tadpole are replaced by lungs as the adult form prepares for life on land. Conclusion: metamorphosis is a complex and regulated biological process that ensures a successful transition from a juvenile form to the adult stage. The process allows organisms to exploit different ecological niches at different life stages, ensuring survival and reproductive success. Hormonal regulation plays a central role in this transformation, with hormones such as juvenile hormone, ecdysone, and thyroid hormones coordinating the timing and progression of metamorphosis. Whether in insects, amphibians, or marine organisms, metamorphosis serves as a fascinating example of developmental plasticity and evolutionary adaptation. You said. Environmental regulation of normal development. ChatGPT said. Environmental regulation of normal development. CSIR Net JRF Life Sciences Lecture. Development is not solely governed by an organism's genetic code. Environmental factors also play a critical role in shaping the course of development. The environment, which includes both internal factors such as hormones and external factors like temperature, light, nutrition, and chemicals, can influence normal development and even alter genetic expression. This interplay between genetics and the environment is known as epigenetic regulation and has far-reaching consequences for the growth, differentiation, and maturation of organisms. Environmental regulation of development is evident across a wide variety of organisms, from plants to animals, and understanding it is crucial for fields such as developmental biology, ecology, and medicine. 1. Overview of environmental regulation. Environmental factors can influence development at multiple stages, from fertilization to adulthood. The concept of plasticity refers to the ability of an organism to alter its developmental trajectory in response to environmental cues. This flexibility allows organisms to survive and thrive in environments. For example, some amphibians can alter their development in response to changes in water temperature, while some plants can alter their flowering time based on light availability. Developmental processes such as cell differentiation, morphogenesis, and organogenesis can be influenced by various environmental cues. In some cases, changes in the environment can lead to phenotypic plasticity, where the same genotype produces different phenotypes depending on the environmental conditions. 2. Temperature and environmental regulation. In both ectothermic animals like amphibians, reptiles, and many fish and plants, temperature is one of the most significant environmental factors influencing development. Temperature can control the timing of developmental processes, such as egg hatching, metamorphosis, and growth rate. Temperature-dependent sex determination (TSD). In certain reptiles like turtles and crocodiles, the sex of the offspring is determined by the incubation temperature of the eggs. Higher or lower temperatures during critical periods of embryonic development lead to the differentiation of either male or female offspring. This is an example of how temperature can directly regulate the expression of sex-determining genes. Developmental rate. In ectotherms, higher temperatures often speed up metabolic and developmental processes. For example, higher temperatures may result in earlier hatching or faster growth rates. In contrast, lower temperatures may delay these processes. In plants, temperature regulates processes like seed germination and flowering. Some plants require specific cold periods fertilization to trigger flowering, while others may require heat to induce germination. 3. Light and environmental regulation. Light is another critical environmental cue that regulates development, particularly in plants and some invertebrates. Light influences a variety of developmental processes, including photoperiodism and circadian rhythms. Photoperiodism in plants. The timing of flowering in many plants is regulated by the photoperiod, which is the relative lengths of day and night. Plants that flower in response to short days are called short day plants, while those that require long days are termed long day plants. The photoperiod affects the expression of genes involved in flowering time and the transition from vegetative to reproductive development. Circadian rhythms. In animals, light regulates circadian rhythms, which control daily cycles of sleep, feeding, and other physiological processes. Light also affects seasonal changes, such as breeding cycles in many animals. 4. Nutritional factors and environmental regulation. Nutritional inputs are essential for normal development. Both the quality and quantity of nutrients available during different stages of development can profoundly affect an organism's growth and maturation. Nutrients such as vitamins, minerals, amino acids, and fatty acids play crucial roles in cellular processes such as DNA replication, protein synthesis, and cell division. In humans and mammals, inadequate nutrition during pregnancy can lead to developmental defects in the offspring. For example, folic acid deficiency during pregnancy can result in neural tube defects in the fetus. Similarly, malnutrition during childhood can lead to stunted growth, cognitive deficits, and weakened immune function. In insects, Drosophila fruit flies, a common model organism, is used to study the role of nutrition in development. Nutritional deprivation or excess can lead to defects in processes like metamorphosis, growth rate, and reproductive output. 5. Hormonal regulation and environmental interaction. Environmental factors can modulate the levels and activity of various hormones that regulate developmental processes. Hormones, which are chemical messengers produced by glands, play a pivotal role in controlling processes like growth, differentiation, and reproductive development. Thyroid hormones. In vertebrates, thyroid hormones such as thyroxine T4 and triiodothyronine T3 are crucial for normal development, especially in amphibians and mammals. The amphibian tadpole undergoes metamorphosis into an adult frog through the action of thyroid hormones, which regulate the development of limbs, resorption of the tail, and the formation of lungs. Environmental endocrine disruptors. Exposure to certain chemicals in the environment, such as pesticides, plasticizers, and industrial chemicals, can interfere with normal hormonal regulation. These endocrine disrupting chemicals, EDCs, may mimic or block the action of natural hormones, leading to developmental abnormalities such as impaired sexual differentiation or neurodevelopmental defects. 6. Chemical factors and environmental regulation. Environmental chemicals can influence development in both positive and negative ways. Phytochemicals, such as those found in plants, can regulate plant growth and development, while toxins and pollutants can impair normal development in both plants and animals. Plant growth regulators (PGRs) in plants, growth regulators like auxins, cytokinins, and gibberellins play key roles in regulating growth and development. These regulators are sensitive to environmental cues like light, temperature, and water availability. Pollution, exposure to pollutants, such as heavy metals or pesticides, can disrupt normal development. For instance, mercury contamination in aquatic environments can lead to developmental defects in fish and amphibians, while air pollution has been linked to respiratory and developmental issues in humans. 7. Epigenetic regulation and environmental influence. Epigenetic mechanisms refer to changes in gene expression that do not involve alterations in the underlying DNA sequence. Environmental factors, such as diet, toxins, and stress, can lead to epigenetic modifications that affect developmental outcomes. These modifications can include DNA methylation, histone modification, and RNA interference. Epigenetic regulation provides a mechanism for organisms to adapt to environmental changes across generations. For example, environmental stressors like toxins or nutrient deprivation can induce changes in the epigenome, leading to altered gene expression patterns that may affect development or even be passed down to subsequent generations. 8. CSIR Net JRF Life Sciences. Previous year questions on environmental regulation of development. CSIR Net JRF June 2020. Which environmental factor influences the timing of flowering in long day plants? Answer: photoperiod, length of daylight. Explanation: long day plants require a minimum length of daylight to trigger flowering, regulated by their sensitivity to the photoperiod. CSIR Net JRF December 2019. What is the role of thyroid hormones in amphibian metamorphosis? Answer: thyroid hormones, particularly T3, initiate metamorphosis in amphibians, leading to the development of limbs and of the tail. Explanation: in amphibians, thyroid hormones regulate the developmental transition from tadpole to adult frog, controlling the formation of limbs and resorption of the tail. CSIR Net JRF June 2018. Which of the following is an environmental factor that can influence the sex of offspring in reptiles? Answer: temperature-dependent sex determination. Explanation: in certain reptiles, such as turtles and crocodiles, the incubation temperature determines whether the offspring will be male or female. Conclusion: environmental regulation of development is a crucial aspect of developmental biology, encompassing a variety of internal and external factors that influence the growth, differentiation, and maturation of organisms. Temperature, light, nutrition, hormonal regulation, and chemical factors all contribute to the modulation of developmental processes. Furthermore, environmental influences can lead to phenotypic plasticity, allowing organisms to adapt to changing environmental conditions. Understanding these regulatory mechanisms is vital not only for developmental biology but also for ecology, evolutionary biology, and medicine, especially with regard to environmental stresses and their impact on organismal health. You said, sex determination. ChatGPT said. Sex determination, CSIR Net JRF Life Sciences Lecture. Sex determination is a critical aspect of developmental biology that dictates the biological sex of an organism. It is controlled by specific genetic, environmental, and hormonal factors that result in the differentiation of male and female organisms. The process of sex determination varies widely across different species, and can be governed by chromosomal systems, environmental factors, or a combination of both. Understanding the mechanisms of sex determination is essential not only in the context of evolutionary biology but also for applications in agriculture, medicine, and conservation. 1. Chromosomal mechanisms of sex determination. In many organisms, sex determination is governed by specific chromosomes that carry the genes responsible for initiating male or female development. These chromosomes are often referred to as sex chromosomes, and different species have evolved different systems of sex chromosome determination. A XY system. In mammals, including humans, the XY sex determination system is the most common chromosomal mechanism. In this system, males possess one X chromosome and one Y chromosome XY. Females possess two X chromosomes XX. The presence of the Y chromosome typically determines male development. The SRY gene, sex-determining region on the Y chromosome, plays a pivotal role in initiating the formation of testes, which secrete male hormones androgens that promote the development of male characteristics. Without the Y chromosome, the default pathway leads to female development. The ZW system. In birds, some reptiles, and some amphibians, the ZW sex determination system is used. In this system, males are homogametic, possessing two ZZ chromosomes. Females are heterogametic, possessing ZW chromosomes. The W chromosome in females carries genes that promote female characteristics, while the Z chromosome plays a role in male development. See haplodiploidy. The ZW system. In birds, some reptiles, and some amphibians, the ZW sex determination system is used. In this system, males are homogametic, possessing two ZZ chromosomes. Females are heterogametic, possessing ZW chromosomes. The W chromosome in females carries genes that promote female characteristics, while the Z chromosome plays a role in male development. See haplodiploidy. In some species of arthropods, particularly in bees, ants, and wasps, a haplodiploid sex determination system is employed. The ZW system. In birds, some reptiles, and some amphibians, the ZW sex determination system is used. In this system, males are homogametic, possessing two ZZ chromosomes. Females are heterogametic, possessing ZW chromosomes. The W chromosome in females carries genes that promote female characteristics, while the Z chromosome plays a role in male development. See haplodiploidy. In some species of arthropods, particularly in bees, ants, and wasps, a haplodiploid sex determination system is employed. Males develop from unfertilized eggs and are haploid, having one set of chromosomes. Females develop from fertilized eggs and are diploid, having two sets of chromosomes. This system is common in social insects, where sex determination influences reproductive roles and social structures. 2. Environmental mechanisms of sex determination. In certain species, sex determination is not dependent on chromosomes but is instead influenced by environmental factors, such as temperature, social interactions, or the presence of certain chemicals. This type of sex determination is seen in many reptiles, fish, and amphibians. A temperature-dependent sex determination (TSD). One of the most well-known examples of environmental sex determination is temperature-dependent sex determination (TSD), observed in many reptiles, including turtles, crocodiles, and some lizards. In these species, the temperature at which the eggs are incubated determines the sex of the offspring. In many turtle species, high incubation temperatures lead to the production of females, while lower temperatures result in males. In some species, the effect of temperature on sex determination is reversed, and different temperature ranges produce different sex ratios. The molecular mechanisms underlying TSD involve temperature-sensitive gene expression pathways that influence the synthesis of sex-determining hormones, which in turn determine the sexual differentiation of gonads. B. Social sex determination. In some species, social structure and environmental cues regulate sex determination. This phenomenon is observed in certain fish, such as clownfish and wrasse. In these, sex determination, CSIR-NET JRF Life Sciences Lecture. Sex determination is a critical aspect of developmental biology that dictates the biological sex of an organism. It is controlled by specific genetic, environmental, and hormonal factors that result in the differentiation of male and female organisms. The process of sex determination varies widely across different species, and can be governed by chromosomal systems, environmental factors, or a combination of both. Understanding the mechanisms of sex determination is essential not only in the context of evolutionary biology but also for applications in agriculture, medicine, and conservation. Chromosomal mechanisms of sex determination. In many organisms, sex determination is governed by specific chromosomes that carry the genes responsible for initiating male or female development. These chromosomes are often referred to as sex chromosomes, and different species have evolved different systems of sex chromosome determination. A XY system. In mammals, including humans, the XY sex determination system is the most common chromosomal mechanism. In this system, males possess one X chromosome and one Y chromosome XY. Females possess two X chromosomes XX. The presence of the Y chromosome typically determines male development. The SRY gene, sex-determining region on the Y chromosome, plays a pivotal role in initiating the formation of testes, which secrete male hormones androgens that promote the development of male characteristics. Without the Y chromosome, the default pathway leads to female development. The ZW system. In birds, some reptiles, and some amphibians, the ZW sex determination system is used. In this system, males are homogametic, possessing two ZZ chromosomes. Females are heterogametic, possessing ZW chromosomes. The W chromosome in females carries genes that promote female characteristics, while the Z chromosome plays a role in male development. See haplodiploidy. In some species of arthropods, particularly in bees, ants, and wasps, a haplodiploid sex determination system is employed. Males develop from unfertilized eggs and are haploid, having one set of chromosomes. Females develop from fertilized eggs and are diploid, having two sets of chromosomes. This system is common in social insects, where sex determination influences reproductive roles and social structures. 2. Environmental mechanisms of sex determination. In certain species, sex determination is not dependent on chromosomes but is instead influenced by environmental factors, such as temperature, social interactions, or the presence of certain chemicals. This type of sex determination is seen in many reptiles, fish, and amphibians. A temperature-dependent sex determination (TSD). One of the most well-known examples of environmental sex determination is temperature-dependent sex determination (TSD), observed in many reptiles, including turtles, crocodiles, and some lizards. In these species, the temperature at which the eggs are incubated determines the sex of the offspring. In many turtle species, high incubation temperatures lead to the production of females, while lower temperatures result in males. In some species, the effect of temperature on sex determination is reversed, and different temperature ranges produce different sex ratios. The molecular mechanisms underlying TSD involve temperature-sensitive gene expression pathways that influence the synthesis of sex-determining hormones, which in turn determine the sexual differentiation of gonads. B. Social sex determination. In some species, social structure and environmental cues regulate sex determination. This phenomenon is observed in certain fish, such as clownfish and wrasse. In these species, environmental factors such as the absence of a dominant male can trigger a female to change sex and become male. In clownfish, all individuals are born male, and the dominant female in a group controls the breeding. If the female is removed, the dominant male transitions into a female, ensuring the reproductive function of the group is maintained. 3. Genetic and epigenetic regulation of sex determination. In addition to chromosomal and environmental factors, epigenetic regulation plays a role in sex determination, particularly in some species where the environment interacts with genetic pathways to influence sex differentiation. Epigenetic mechanisms involve changes in gene expression without altering the DNA sequence, such as DNA methylation, histone modification, and non-coding RNA involvement. These modifications can be influenced by environmental factors like diet, temperature, or toxins, potentially leading to changes in sex determination pathways. 4. Sex determination in human disorders. In humans and other mammals, deviations from the typical XY chromosomal pattern can lead to sex chromosome disorders. These include conditions such as Turner syndrome. Individuals with 45, X have only one X chromosome and no second sex chromosome. They develop as females but often have developmental issues, including short stature and infertility. Turner syndrome. Individuals with 45, X have only one X chromosome and no second sex chromosome. They develop as females but often have developmental issues, including short stature and infertility. Klinefelter syndrome. Males with 47, XXY chromosomes have an extra X chromosome. These individuals may have underdeveloped male characteristics and infertility. Androgen insensitivity syndrome. ICE individuals with XY chromosomes may have a normal male genotype but fail to respond to androgens, resulting in the development of female external characteristics despite the presence of male internal organs. 5. CSIR Net JRF Life Sciences. Previous year questions on sex determination. CSIR Net JRF June 2020. What is the role of the SRY gene in sex determination in humans? Answer. The SRY gene on the Y chromosome triggers male development by initiating the formation of testes. The testes produce male hormones, androgens, that promote male differentiation. CSIR Net JRF December 2019. Which of the following organisms exhibits temperature-dependent sex determination TSD? Answer. Turtles. Explanation. In many species of turtles, the temperature at which the eggs are incubated determines the sex of the offspring. CSIR Net JRF June 2018. Which of the following is true regarding sex determination in Drosophila? Answer. In Drosophila, sex determination is based on the ratio of X chromosomes to autosomes. Females are XX, while males are XY. Explanation. The sex in Drosophila is determined by the X-A ratio, where a ratio of 1.0XX leads to female development, and a ratio of 0.5XY leads to male development. Conclusion. Sex determination is a fundamental process that dictates the biological sex of an organism, and it can be regulated by a variety of mechanisms, including chromosomal systems, environmental factors, and epigenetic regulation. The diversity in sex determination mechanisms across different species highlights the adaptability and evolutionary significance of this process. In mammals, the XY system predominates, while in other organisms like reptiles and fish, environmental factors such as temperature and social structure can govern sex determination. Understanding these mechanisms is essential for comprehending both normal development and the effects of environmental and genetic disruptions. Search. Rhizo. CSIR Net JRF Life Sciences. DNA replication, prokaryotes and eukaryotes. DNA replication is a biological process through which a cell copies its entire genome before cell division. This semi-conservative mechanism ensures that each daughter cell receives an identical morphogenesis and organogenesis in plants, ChatGPT said. Here is your CSIR Net JRF Life Sciences lecture on the topic, morphogenesis and organogenesis in plants, written in a paragraph-only format, fully packed with maximum level detail, no tables, and includes previous year CSIR questions with answers in year mentioned. This lecture is designed to be your only coaching material, complete, advanced, and aligned with JRF standards. Morphogenesis and organogenesis in plants, CSIR Net JRF Life Sciences. Morphogenesis in plants refers to the biological processes that govern the shape, structure, and spatial distribution of organs and tissues during development. Organogenesis is a specialized subset of morphogenesis that focuses on the formation of specific organs such as roots, shoots, and leaves from undifferentiated cells or tissues. In plants, unlike animals, development is highly plastic and indeterminate, with the capacity for continuous organ formation throughout life due to the persistent activity of meristems. The shoot apical meristem (SAM) and root apical meristem (RAM) are the primary sites where morphogenesis initiates. These meristems are composed of pluripotent cells that undergo tightly regulated cell division, expansion, and differentiation, governed by hormonal signaling and transcriptional control. The hormonal regulation of morphogenesis is primarily driven by auxins and cytokinins. Auxin, especially indole 3-acetic acid (IAA), plays a central role in establishing polarity, apical dominance, and pattern formation by regulating gene expression through AUX, IAA, and ARF transcription factor pathways. It is asymmetrically distributed via kin proteins, auxin efflux carriers, creating local auxin maxima that direct organ primordial formation. Cytokinins promote cell division and are essential for shoot initiation. The balance between auxin and cytokinin concentrations determines the developmental fate of cells. A high auxin to cytokinin ratio induces root formation, while a high cytokinin to auxin ratio promotes shoot formation. This hormonal interplay forms the basis for in vitro organogenesis, a fundamental technique in plant tissue culture. In vitro morphogenesis can proceed through two primary pathways, organogenesis and somatic embryogenesis. In direct organogenesis, organs develop directly from explant tissues, often in response to external phytohormones. In indirect organogenesis, a callus phase to differentiated mass of cells precedes organ formation, and redifferentiation is induced by altering hormonal conditions. Somatic embryogenesis involves the formation of embryo-like structures from somatic cells, capable of developing into entire plants. This process mirrors zygotic embryogenesis but occurs without fertilization. Both organogenesis and somatic embryogenesis are governed by transcription factors like WSCHEL, WOOS, shoot MERISTM, LESS, STM, and leafy cotyledon LEC. WOOS, for instance, maintains stem cell identity in the SAM and is necessary for shoot regeneration. LEC genes are crucial for the induction of embryogenic fate during somatic embryogenesis. At the molecular level, morphogenesis involves gene networks such as the NOX genes in shoot meristem maintenance and YAVI and HTZIPII genes in leaf polarity. Epigenetic mechanisms like DNA methylation and histone modifications also regulate gene expression patterns during morphogenesis. The role of microRNAs, e.g., miR165-166 targeting HTZIPII, adds another layer of regulation, fine-tuning the expression of developmental genes spatially and temporally. Positional information and cell-cell communication are fundamental for patterning. Plants utilize both chemical signals and mechanical cues to direct growth and differentiation. Plasmodesmata-mediated signaling, small peptides, e.g., clavata-3, and receptor-like kinesis contribute to the maintenance of meristematic zones and organ positioning. Morphogenesis is also sensitive to environmental cues such as light and gravity, with photoreceptors and statoliths guiding differential growth responses, contributing to organ orientation and adaptive morphogenesis. Thus, morphogenesis and organogenesis in plants are dynamic processes integrating hormonal cues, genetic programs, and environmental signals to produce diverse and functional plant structures. Their study not only sheds light on fundamental plant development but also underpins applications in agriculture, forestry, and biotechnology, particularly in the propagation of elite genotypes and genetic transformation. Previous year CSIR net questions from this topic. CSIR net June 2014. Question. In a tissue culture experiment, which hormonal combination would most likely induce shoot formation from a callus? A. High auxin, low cytokinin. B. High cytokinin, low auxin. See equal concentrations of auxin and cytokinin. D. Number hormones. Answer. B. High cytokinin, low auxin. This question tests the hormonal basis of in vitro morphogenesis. CSIR net December 2015. Question. A mutation in the WSCHEL gene in Arabidopsis would most likely result in which phenotype? A. Loss of root hairs. B. Loss of shoot apical meristem. C. Extra leaves. D. Abnormal flower color. Answer. B. Loss of shoot apical meristem. WUS is required for the maintenance of stem cells in the SAM. CSIR net June 2017. Question. Somatic embryogenesis differs from organogenesis primarily in which of the following ways? A. Requires higher cytokinin levels. B. Involves zygote formation. C. Forms bipolar structures capable of forming entire plants. D. Occurs only in monocots. Answer. C. Forms bipolar structures capable of forming entire plants. Somatic embryos mimic zygotic embryos and develop into full plants. Would you like to continue with the next topic, e.g., plant hormones, photoperiodism, or a topic of your choice? You said. Organization of shoot apical meristem. ChatGPT said. Here is your CSIR net JRF Life Sciences high quality, paragraph based, fully detailed lecture on organization of shoot apical meristem SAM. This is crafted specifically for JRF level success, using previous year question trends, and includes 2 to 3 CSIR net questions with answers in years mentioned. No free space is wasted. This is your complete and only coaching material. Organization of shoot apical meristem SAM CSIR net JRF Life Sciences. The shoot apical meristem SAM is the primary site of post-embryonic aerial organ development in plants, playing a central role in morphogenesis and organogenesis. It consists of a dome-shaped group of pluripotent stem cells located at the tip of the shootus. These cells retain the capacity for continuous division, providing new cells for the formation of leaves, stems, and floral structures while maintaining their own undifferentiated state. The SAM is a highly organized structure with distinct functional and histological zones, governed by intricate molecular signaling networks, positional information, and hormonal cues. SAM organization is understood in 2 complementary frameworks: histological zonation and functional zonation. The histological zonation is based on patterns of cell division and ring. It divides the SAM into 3 clonally distinct layers. L1 layer, the outermost single cell layer that divides only anticlinally perpendicular to the surface. It gives rise to the epidermis. L2 layer, the second layer that also divides anticlinally but gives rise to subepidermal tissues, including mesophyll and ganglionic tissue. L3 layer, the innermost layer with divisions in all planes, contributing to the inner tissues of the stem and vascular tissues. The functional zonation divides SAM into 3 major regions. The central zone CZ is located at the tip of the meristem and consists of slowly dividing stem cells that act as a reservoir of undifferentiated cells. The peripheral zone PZ surrounds the central zone and is more mitotically active, contributing cells for organ primordia, leaf, flower initiation. The rib meristem or pith meristem lies beneath the CZ and contributes to the central stem tissues. The homeostasis of the SAM depends on a fine-tuned balance between stem cell maintenance and differentiation, governed by a feedback loop between 2 critical genes, WSCHEL, WUS, and Clavata CLB. WUS, expressed in the organizing center beneath the CZ, promotes stem cell identity by non-cell autonomous signaling to the CZ. CLB3, a small peptide secreted by stem cells, restricts WUS expression by activating the CLB1, CLB2 receptor complex, forming a negative feedback loop that ensures stable stem cell number. Mutations in WUS lead to the premature differentiation of the meristem, whereas CLB mutants result in enlarged meristems due to excessive stem cell accumulation. The SAM is also under hormonal regulation. Cytokinins promote stem cell proliferation by inducing WUS expression. Auxin, though typically associated with organ initiation, is kept at low levels in the CZ to prevent premature differentiation, whereas auxin maxima at the periphery PZ promote leaf and floral organ initiation. Gibberellin, SCA, and brasinosteroids also modulate SAM activity, often by regulating boundary genes like cup-shaped cotyledon that define organ separation. Transcription factors are central to SAM identity and organization. In addition to WUS, shoot MERISTEMLESS, STM maintains the meristematic state by preventing differentiation. STM mutants exhibit failure in meristem formation, demonstrating its essential role. Other factors include NOX genes, e.g., KNAT1, KNAT2, that repress gibberellin biosynthesis to keep cells undifferentiated. The asymmetric LEADS1-AS1 gene interacts antagonistically with NOX to ensure proper leaf development, indicating crosstalk between meristem and organ identity pathways. The SAM also exhibits dynamic patterning guided by auxin transport via PIN proteins. Auxin gradients influence where new primordia will form on the periphery, a process controlled by phylotactic patterns, e.g., spiral, opposite, whorl, which are critical for the spatial distribution of organs. The phyllotaxy is ultimately a result of auxin-mediated inhibition fields and transport canalization. In summary, the shoot apical meristem is a highly dynamic, multi-layered, and multifunctional structure integrating cell division patterns, hormonal gradients, transcriptional networks, and feedback mechanisms to ensure the continuous and controlled development of above-ground plant organs. Its study provides crucial insight into the principles of plant development and has profound implications for crop improvement and plant tissue culture. Previous year CSIR net questions from this topic. CSIR net deck 2016. Question: which of the following gene products is known to maintain stem cell identity in the shoot apical meristem? A. Leafy. B. Clavata1 circuit WSCHELDLEC2. Answer: C. Wuschel. Wuschel is essential for stem cell maintenance in SAM. CSIR net June 2013. Question: in Arabidopsis, overexpression of Clavata3 leads to which of the following phenotypes? A. Loss of root meristem. B. Enlarged shoot apical meristem. C. Decreased stem cell population in SAM. Diffusion of floral organs. Answer: C. Decreased stem cell population in SAM. CLB3 restricts WUS activity. Overexpression reduces stem cells. CSIR net June 2018. Question: in an experiment, Arabidopsis seedlings are treated with a cytokinin inhibitor. What is the expected effect on the shoot apical meristem? A. Enlargement of SAM. B. Loss of WUS expression and stem cell activity. C. Increased leaf initiation rate. D. Formation of ectopic roots. Answer: B. Loss of WUS expression and stem cell activity. Cytokinin is required to induce and maintain WUS expression. Would you like to continue with root apical meristem, auxin transport, or another next topic? Just let me know. You said. Organization of root apical meristem. ChatGPT said. Here is your CSIR net JRF Life Sciences full length, paragraph type, maximum detail lecture on organization of root apical meristem RAM tailored specifically to JRF level requirements. This note is written with zero wasted space, covers content from base to advanced level, includes key previous year question areas, and ends with 2 to 3 previous CSIR net questions with answers in years mentioned. No tables, only enriched theory. Organization of root apical meristem RAM CSIR net Life Sciences. The root apical meristem (ram) is a post-embryonic, indifferentiated tissue at the tip of the root responsible for continuous root growth. Unlike the shoot apical meristem, which gives rise to aerial organs, the ram is responsible for generating root tissues in a highly organized and stable pattern. The ram maintains a balance between stem cell self-renewal and differentiation of daughter cells to ensure persistent root elongation and development. It is a key model for understanding cellular organization, positional signaling, and stem cell niche regulation in plant developmental biology. At the heart of the ram is the quiescent center QC, a group of slowly dividing or metabolically inactive cells that serve as a reservoir to maintain the surrounding actively dividing stem cells. The QC is essential for the identity and maintenance of stem cells and was first described by Klaus in apes. It is characterized by low meiotic activity, low DNA synthesis, resistance to radiation, and unique expression of molecular markers such as WOX5, Wechsel-related homeobox5. WOX5 is functionally analogous to Wus in SAM and is essential to maintain the surrounding initial cells in an indifferentiated state. Ablation of the QC or loss of WOX5 expression causes premature differentiation of root stem cells, showing the central regulatory role of the QC. The ram is organized in a radial symmetry, and tissues are formed in concentric layers from the divisions of stem cells surrounding the QC. These stem cells, also called initial cells, give rise to the three primary tissue systems of the root. The protoderm, which forms the epidermis. The ground meristem, which gives rise to the cortex and endodermis. The procambium, which forms the vascular cylinder, xylem and phloem. The columella initials, located just below the QC, give rise to the columella root cap, a structure critical for gravity sensing. The lateral root cap initials flank the columella initials and generate the lateral root cap. These cap structures protect the ram as it pushes through the soil and secrete mucilage to ease root penetration. Ram organization can be of two types, closed meristem and open meristem. In closed meristems, the initials for you said, continue ChatGPT said. In closed meristems, the initials for different tissue types are arranged in distinct groups with clear boundaries. For example, separate initials for root cap, epidermis, cortex, and vascular tissues. This organization is observed in many monocots like maize. In contrast, open meristems have no clear separation among the initials, and all tissues arise from a common group of initials. This type is common in dicots such as Arabidopsis. Despite these differences in structural demarcation, the underlying genetic control of ram identity and maintenance is evolutionarily conserved across species. The quiescent center QC plays a pivotal signaling role. It maintains adjacent initials in a stem cell-like state through non-cell autonomous signaling. The gene WOX5, specifically expressed in the QC, represses differentiation and promotes stem cell fate in columella initials. Genetic studies show that loss-of-function mutations in WOX5 cause stem cells to undergo premature differentiation, especially in the columella region. QC cells are also responsible for reactivating stem cells after stress or damage. This regeneration capacity underscores their importance as a backup reservoir for stem cell reconstitution. Auxin gradients play a fundamental role in establishing and maintaining RAM. A peak of auxin concentration exists in the QC region and is established by polar auxin transport through PIN-FORMED PIN proteins. PIN1, PIN2, and PIN4 are essential for establishing auxin maxima in the root tip. Auxin controls both the patterning and maintenance of RAM by regulating the expression of PLT plethora of genes. These PLT transcription factors act as master regulators of root stem cell identity and are expressed in a concentration-dependent gradient. High levels near the QC maintain stemness, while reduced levels away from the QC promote cell differentiation. Loss of PLT genes causes root meristem exhaustion and impaired root growth. RAM maintenance also involves SCR, scarecrow, and SHR-SHORTROOT genes, which control radial patterning of the ground tissue. SHR is expressed in the steel but moves to adjacent cell layers and activates SCR to specify endodermal identity. This movement also contributes to stem cell niche specification, especially in the cortex and endodermis initials. Their interaction is crucial for the asymmetric cell divisions that establish the radial tissue layers of the root. The CYCD type cyclins, CDKs, and their inhibitors like KRP proteins regulate the cell cycle in RAM. The activity of these regulators is tightly controlled spatially and temporally. While cells in the QC remain mostly arrested in the G1 phase to preserve genomic integrity, their surrounding initials cycle rapidly to contribute to new cells. Stress conditions, such as genotoxic damage or nutrient deprivation, trigger a further slowdown of QC division to protect meristem longevity. RAM is also influenced by external signals, such as environmental stress, nutrient levels, and hormones like cytokinin, which acts antagonistically to auxin. Cytokinin promotes differentiation and restricts RAM size by negatively regulating PLT expression and by activating the ARR, Arabidopsis response regulator, family of transcription factors. The antagonism between auxin and cytokinin defines a developmental balance between proliferation and differentiation in the RAM, a principle that echoes in many developmental systems. Recent research highlights the involvement of microRNAs, like near 165-166, in regulating HDs3 transcription factors that affect xylem patterning and radial identity. Moreover, epigenetic mechanisms, including chromatin remodeling and DNA methylation, influence RAM function by controlling gene expression dynamics in stem cell niches. In conclusion, the organization of the root apical meristem involves a coordinated interaction of structural zones QC, stem cells, initials, molecular regulators WOX5, PLT, SCR, SHR, PINs, and hormonal gradients auxin, cytokinin. Its precise regulation ensures a continuous supply of root tissues and confers plasticity to respond to environmental conditions. Understanding RAM not only deepens our grasp of plant development but also offers practical implications in agriculture, stress resistance, and root engineering for crop improvement. Previous year CSIR-NET questions. RAM topic CSIR-NET June 2014. Question. WOX5 expression in Arabidopsis root apex is critical for maintaining which of the following? A lateral root formation. B vascular cambium activity. C stem cell identity in root tip. The endodermis development answer. C stem cell identity in root tip. WOX5 maintains stem cells in the QC of the root apical meristem. CSIR-NET Deck 2016. Question. What is the consequence of a mutation in the SHR gene in Arabidopsis roots? A excessive root hair formation. B absence of endodermal layer. C disruption of shoot phyllotaxy. D enlarged quiescent center answer. B absence of endodermal layer. SHR is essential for endodermis specification and movement into cortex. CSIR-NET June 2018. Question. Inhibition of PIN proteins in the root tip leads to enhanced RAM activity. B diffused auxin gradient and loss of RAM organization. C increased cytokinin signaling. D root cap elongation. Answer. B diffused auxin gradient and loss of RAM organization. Pins establish auxin maxima at the QC. Their inhibition disrupts root meristem maintenance. Would you like the next topic, like lateral root formation, role of auxin in root patterning, or move to another section like photosynthesis or cell signaling? Let me know your next topic and I'll prepare it just like this. Full power, no space wasted. You said, shoot development, ChatGPT said. Here is your CSIR-NET JRF Life Sciences full-length lecture on shoot development, crafted to the highest possible quality, fully tailored for CSIR-NET JRF level. The lecture is written in a dense, paragraph-only format, using maximum word limit, and includes 2 to 3 CSIR-NET previous year questions with year and answer mentioned. No tables, no bullets, no wasted space, only exam-worthy information from base to advanced level. Shoot development, CSIR-NET Life Sciences. Shoot development is a complex and finely regulated process that begins during embryogenesis and continues throughout the plant's life. The shoot system includes the stem, leaves, and reproductive structures like flowers and fruits. The central engine of post-embryonic shoot development is the shoot apical meristem, SAM, a dome-shaped mass of pluripotent stem cells located at the apex of the shoot. This meristem maintains a delicate balance between self-renewal of stem cells at its core and the initiation of organ primordia at its flanks, leading to the continuous production of lateral organs such as leaves, branches, and floral meristems. The SAM is organized into two main domains, a central zone CZ containing slowly dividing stem cells and a peripheral zone PZ where cells divide more actively and are recruited for organogenesis. Beneath these lies the rib meristem RM, which contributes to the internal tissues of the stem. The homeostasis of these zones is tightly regulated by key genetic networks, hormonal cues, and feedback signaling pathways. One of the master regulators of shoot meristem maintenance is WSCHEL, WOOS, a homeobox gene expressed in a group of organizing center cells beneath the central zone. WOOS maintains the indifferentiated state of the overlying stem cells through non-cell autonomous signaling. WOOS positively regulates CLBA3, a small secreted peptide produced by stem cells, which in turn activates the CLB1, CLB2 receptor kinase complex to repress WOOS expression. This WOOS CLB feedback loop is essential for the size regulation of the meristem. Mutations in CLB genes result in enlarged SAMs due to excessive stem cell accumulation, while loss of WOOS leads to premature differentiation and meristem termination. The fine-tuning of this loop ensures that stem cell proliferation and differentiation remain in balance. Phytohormones play crucial roles in shoot development. Cytokinin promotes stem cell proliferation and positively regulates WOOS expression, while auxin is involved in organ initiation at the meristem periphery. Local auxin maxima, established by polar auxin transport through PIN-FORMED, PIN-efflux carriers, specify the sites of organ initiation. The transcription factor MONOPTEROS-MP and ARF auxin response factor mediates auxin signaling during leaf and flower primordia formation. The transition from vegetative to reproductive development is also regulated by hormonal and environmental inputs that trigger the expression of leafy LFY and petal-1AP1, which act as floral meristem identity genes. These transitions involve the reprogramming of the shoot apex and the suppression of vegetative pathways by floral inducers such as flowering locus TFT. The initiation of leaves, a critical component of shoot development, occurs at regular intervals due to a process known as phyllotaxy, which is governed by the spatial patterning of auxin distribution. The PIN1 auxin efflux transporter redirects auxin flow to sites of incipient primordia, where auxin accumulation initiates organ outgrowth. Once a primordium is initiated, it becomes an auxin, depleting auxin in surrounding areas and preventing adjacent primordia formation, thereby maintaining phyllotaxic spacing. The identity and polarity of the developing leaf are established by the differential expression of NOX, knotted one-like homeobox genes in YABI, KANADI, and fabulosa phagocytic transcription factors. NOX genes are expressed in the SAM but are repressed in leaf primordia to allow determinate organ development. Ectopic expression of NOX genes in leaves leads to lobed or compound leaf forms. The formation of axillary meristems, which give rise to branches, is another fundamental aspect of development. These meristems originate from cells of the leaf axil that retain meristematic competence. The gene lateral suppressor LAS in Arabidopsis and its rice ortholog molecule 1, MOC1, are essential for the initiation of axillary meristems. Their expression is regulated by auxin cytokinin crosstalk and positional cues from the main SAM. Once initiated, axillary meristems can either remain dormant or grow into branches depending on environmental and endogenous signals. Strigolactones, a class of branching inhibitors, act through MAX more axillary growth genes to repress bud outgrowth, integrating resource availability and hormonal signals. In later stages of shoot development, the stem elongates through intercalary meristem activity and the vascular system differentiates, establishing connectivity between roots and leaves. The transcriptional regulation of vascular patterning involves HDZIP3 family genes and is sensitive to auxin fluxes. In reproductive development, the inflorescence meristem forms from the shoot apex and eventually gives rise to floral meristems. The ABC model of floral organ identity, regulated by NADS box genes, is activated during this transition and determines the identity of sepals, petals, stamens, and carpels. At the cellular level, shoot development requires precise regulation of cell division, expansion, and differentiation. These processes are governed by cell cycle regulators, microRNAs, chromatin remodelers, and cell-to-cell communication. Plasmodesmata, the cytoplasmic channels between plant cells, play a critical role in transporting developmental signals, including transcription factors and RNAs that coordinate patterning across tissues. Epigenetic regulation, including histone modifications and DNA methylation, also modulates gene expression during shoot development. For example, the polycomb repressive complex 2 (PRC2) silences flowering repressors like flowering locus C (FLC) to promote the floral transition. Shoot development is highly plastic and responsive to environmental conditions such as light, gravity, temperature, and nutrient status. Photoreceptors like phytochromes and cryptochromes mediate light-dependent shoot elongation and leaf development, integrating external cues into developmental pathways. Photokaryotic control through constants, CO, and FT proteins links day length to flowering time, which is a key agricultural trait. In summary, shoot development is a continuous, modular, and environmentally responsive process regulated by a network of transcription factors, phytohormones, epigenetic modifiers, and positional signals. The coordination between meristem maintenance, organ initiation, and differentiation ensures proper shoot architecture, vegetative growth, and reproductive success. A comprehensive understanding of shoot development is not only fundamental for developmental biology but also has direct implications in crop improvement, yield enhancement, and plant architecture manipulation. Previous CSIR-NET questions. Shoot development CSIR-NET June 2015. Question: which of the following gene pairs functions antagonistically to regulate shoot apical meristem size in Arabidopsis? At LFY and AP1. B-WOOS and CF3 circa FT and FLCD YABI and NOX answer. B-WOOS and CLB3. They maintain meristem size through a negative feedback loop. CSIR-NET Deck 2017. Question: PIN1 mutants in Arabidopsis show severe defects in the root hair elongation. D-leaf vascular patterning C floral organ identity D phyllotaxic pattern of leaves answer. D phyllotaxic pattern of leaves. PIN1 is critical for auxin-mediated organ positioning in the SAM. CSIR-NET June 2020. Question: loss of function mutation in the NOX gene in the SAM results in an increased cell death. B loss of leaf polarity. C premature differentiation and loss of meristem maintenance D enhanced cytokinin biosynthesis answer. C premature differentiation and loss of meristem maintenance NOX genes are required to maintain indeterminacy in the SAM. Let me know the topic: floral meristem identity, root patterning, totipotency, light signaling, transgenic plants, or anything else. I'll deliver it in the same full-strength format. You said root development. ChatGPT said. Here is your CSIR-NET JRF Life Sciences full syllabus lecture on root development, crafted to be dense, high level, and tailored for CSIR-NET based on previous year question trends. It is written in paragraph format only, using the maximum word limit, and includes 2 to 3 solved CSIR-NET question year and answer. This lecture is prepared with zero wasted space, serving as your sole coaching material. Root development, CSIR-NET JRF Life Sciences. Root development in higher plants is a tightly coordinated and highly conserved process that begins during embryogenesis and continues post-embryonically under both intrinsic genetic control and environmental cues. It is initiated by the specification of the hypophysis, a cell in the early embryo that gives rise to the quiescent center QC and the root apical meristem RAM. The RAM maintains continuous growth of the primary root through a regulated balance between cell division in the meristematic zone, elongation in the transition zone, and differentiation in the maturation zone. The RAM is organized into three major components. The quiescent center, a group of mesotically less active cells that serve as a reservoir of undifferentiated stem cells. The initial cells surrounding the QC that give rise to different tissues like the epidermis, cortex, endodermis, and vascular tissues, and the root cap initials, which produce cells for the protective root cap. Hormonal signaling, particularly auxin, is central to root development. Auxin establishes a concentration gradient with a maximum at the root tip, maintained by polar auxin transport mediated by PIN-FORMED, PIN, and auxin-resistant 1-OX1 transporters. This gradient is essential for maintaining the identity of the QC and for initiating root patterning. The plethora of PLT gene family acts downstream of auxin and forms a gradient-like expression that defines different zones of the root. High levels of PLT in the tip maintain stem cell identity, while decreasing levels in the elongation and differentiation zones allow cells to progress through developmental stages. This zonation is critical for the proper organization of root tissues and overall root architecture. Root development also depends on the short ROT SHR and scarecrow SCR genes, which regulate radial patterning of the root. SHR is produced in the stele and moves into the surrounding endodermal cells, where it activates SCR. Together, they promote paraclinal cell division that establishes the cortex and endodermis layers. SHR also plays a role in maintaining stem cell identity by restricting QC identity to the appropriate domain. The WOX-WUSCHEL related homeobox gene family, particularly WOX5, functions analogously to WUS and the SHUG and is specifically expressed in the QC. WOX5 maintains the surrounding stem cells in an undifferentiated state and represses differentiation promoting genes. The initiation of lateral roots (LRs) is a key aspect of post-embryonic root development. LRs originate from the paracycle, specifically from xylem pole paracycle cells that re-enter the cell cycle. This process is tightly regulated by auxin maxima, local redistribution of auxin by PIN3 and PIN7, and transcriptional activation of GATA23, LBD16, and LBD29, which trigger founder cell identity and division. The development of lateral roots proceeds through tightly coordinated stages: founder cell specification, anticlinal and paraclinal divisions, primordium formation, and emergence through overlying tissues. OX-IAA proteins and ARFs, auxin response factors, also play significant roles in this process. In particular, IAA14 SLR represses ARF7 and ARF19 in the absence of auxin, while its degradation upon auxin binding leads to LR initiation. Root hair development is another specialized differentiation event in the root epidermis and is important for increasing root surface area and nutrient absorption. Root hairs are derived from trichoblasts, and their fate is determined by positional information related to underlying cortical cells. Key regulators include WER werewolf, which represses hair cell fate, and CPC caprice, which promotes it. The transcription factor root hair defective 6 RHD6 is crucial for hair initiation, while RSL root hair defective 6 like proteins promote elongation. Root hair initiation and elongation are further influenced by hormonal signals, especially auxin, ethylene, and racinosteroids. Environmental factors such as phosphate availability, gravity, and water stress also significantly affect root development. Under low phosphate, primary root growth is inhibited while lateral root and root hair development are enhanced, an adaptive strategy controlled by PHR1, PHL1, and ALMT1 pathways. Gravitropism, the directional growth of roots in response to gravity, is regulated by auxin redistribution facilitated by PIN3 and PIN7 in the columella cells of the root cap. These changes lead to differential cell elongation and root bending. The metanosensing of soil hardness and hydrotropism are also vital cues influencing root development and plasticity, allowing plants to adapt to diverse soil environments. In monocots, such as rice and maize, root systems are fibrous and consist of adventitious roots, crown roots, and lateral roots, forming post-embryonically. These roots arise from the stem tissue and are crucial for anchorage and nutrient uptake. The genetic control of adventitious root formation involves WOX11, ARF7, LBD29, and cytokinin signaling pathways, revealing conservation and divergence in root development mechanisms across species. Root architecture, shaped by these developmental processes, is crucial for plant productivity and is a target for crop improvement under stress conditions. Epigenetic regulation, such as histone modifications, chromatin remodeling, and microRNAs, plays an essential role in the spatial temporal control of gene expression during root development. For example, MIR390TAS3ARF pathway fine-tunes auxin responses during LR formation, while chromatin modifiers like SWI SNF regulate the expression of PLT and WOX genes. Stem cell niche maintenance, cell lineage specification, and tissue patterning are therefore outcomes of the integrated action of transcriptional regulators, mobile signals, hormonal gradients, and epigenetic controls. In conclusion, root development is a dynamic, tightly orchestrated developmental program that integrates internal genetic regulators and external environmental signals. It involves the coordination of cell division, elongation, and differentiation processes, beginning from embryonic patterning to the formation of a functional and adaptive root system. Understanding these processes at the molecular and cellular levels is essential not only for basic plant developmental biology but also for agricultural innovations in root trait engineering for improved water and nutrient uptake under variable conditions. Previous year CSIR net questions. Root development CSIR net deck 2016. Question: which one of the following gene pairs is involved in radial patterning of the root? A WUS and CLV3. BSHR and SCR circa LFY and AP1 DPIN1 and PID answer. BSHR and SCR. They regulate cortex and endodermis specification through movement and activation. CSIR net June 2017. Question: WOX5 is expressed in which of the following zones in the Arabidopsis root? A elongation zone. B differentiation zone. C quiescent center. D vascular cylinder answer. C quiescent center. WOX5 maintains stem cell identity in the root meristem. CSIR net deck 2019. Question: mutation in SLR IAA14 leads to which of the following root phenotypes in Arabidopsis? A enhanced root hair elongation. B abnormal root gravitropism. C inhibition of lateral root formation. D increased primary root length answer. C inhibition of lateral root formation. SLR IAA14 inhibits ARF7 ARF19, blocking lateral root initiation in the mutant. Let me know the next topic: floral development, phylotaxy, totipotency, or molecular basis of organogenesis, and I will provide it in the same perfect format. You said, leaf development. ChatGPT said.